Hello. 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 Good evening. Good evening, one and all. ಎಲ್ಲೈತು गुड इवनिंग इज माय वॉइस इज क्लियर बताओ कमेंट में लाइट ए ಇಷ್ಟನಾ yeah good evening sir voice is low little bit low sir okay
Is it low for everyone? Just confirm once, like if the voice is low for everyone. एक बार confirm कर दो. Start करते हैं. Not clear, sir. So voice is not clear. देखो क्यों हो रहा भाई ये? No. एक minute. So the voice is a bit strange. How about now? Is it fine? अभी voice ठीक है? एक बार तुम बात करना भाई.
hello is my voice is clear
अभी clear है.
कंफर्म कर दो ठीक है
तो इसको थोड़ा स्क्रीन ब्लैक आ रहा है क्या? चेहरा ब्लैक है?
no
थोड़ा इसको गोरा कर दे. अभिषेक सर का थोड़ा व्हाइट बना दे.
अरे भाई मैं वाइट ही हूँ
व्हाइट साला काला है तू. और काला कर दे या व्हाइट कर दे इसको.
चलो पढ़ें और
अभी देखना मोटू काला काला नहीं सफेद. ये क्या बोलते सफेद ये क्या है? व्हाइट हो गया थोड़ा.
remove the screen from detergent अरे भाई चिंचो it's too much
अभी ठीक है. ब्राइटनेस इंक्रीज. अभी
okay
ठीक है ना?
everything is fine I think
पहले से बेटर है ना अभी? सो जस्ट शो द टेक्स्ट वंस. अभी सब कुछ क्लियर है स्क्रीन पे? एवरीथिंग इज क्लियर? जस्ट कंफर्म वंस. जस्ट गिव अ थम्स अप. अरे 2 stars. oh my god. okay फाइन. ठीक है.
okay we were not going to waste time today we will just finish again there will be some 20-25 questions in this particular ICAR PG 2023 exam okay again there will be so many repetitive questions okay no problem we will see what are those the repeated questions also and we will try to see what are the new questions they have asked in this particular 2023 shift. Sir, please conduct session for agriculture microbiology also. Yeah, we will see after this breeding and genetics. Okay, we will try to cover first the important main subjects. Okay, anyway you already know about me. Okay, this is the slide for my you guys for my information. You can just see the first question. Okay, which of the following activities is not practical? So, okay, sorry, not practiced in the bulk breeding method. Bulk breeding method. Not practiced. अंकुर बता रहा है सी डी. अरे डी क्यों बता रहा है? बल्क ब्रीडिंग में हम पेडिग्री रिकॉर्ड मेंटेन नहीं करता है, ठीक है. सी सी डी डी विजयाली क्यों डी लगा रहा है आप? अनुराते भी डी लगा रहा है, ठीक है. आंसर इज सी. ऑफ सीजन नर्सरीज एंड ग्रीन हाउस फैसिलिटीज आर यूज्ड टू advanced generation. Okay, it can be used in SSD or backcross breeding method. Okay, since in backcross breeding method we just need to advance generation so that we can efficiently use off-season nurseries and greenhouse facilities. But in case of bulk breeding method, we need environment to act upon it. Okay, natural selection. सुना है ना natural selection bulk breeding में, इसलिए हम we are not going to use off-season nurseries and greenhouse facilities in case of bulk breeding method. What exactly we will do in bulk breeding method? First, we will create a variation. Without create variation, it is just like mass selection. Okay, the same procedure we will use by creating variation in the bulk breeding method. First, we will cross two parents. Okay, then we will get F1, then F2. F2 onwards, we will try to bulk the selected material up to F7, F8. Okay, when the favorable environment will appear, okay, for example, any disease or any pest will occur, so that we can select the best possible plants. Okay, now here at this particular bulk breeding method, okay, we can wait for an opportunity for the selection, and also we can give sorry for the interruption. In backcross breeding, there will be two main advantage. Okay, we will wait for opportunity for selection, and also we also will give for nature to select the plants. Okay, natural selection. These are the two important things you should remember in backcross breeding. Yeah. If the values of phenotypic and environmental variance are 6.8, 1.2 respectively, the heritability, broad sense they are asking, of the trait would be the heritability. Batao. There you should see, okay, in what form they are asking. They are asking in form of percentage. Okay, you should see for the percentage. Okay, they have given phenotypic variance, that is 6.8. They have given environmental variance 1.2. B. Yeah, okay, we will see. So if you need genetic variance, so you should minus 6.8 minus 1. You should minus 6.8. Sorry. Your phenotypic variance from environmental variance 6.8 minus 1.2. So you will get 5.6. So what about your broad sense heritability formula? Genotypic variance divided by phenotypic variance. Your genotypic variance will be 5.6. Your phenotypic variance will be 6.8. Okay, if you do the calculation, you will get 82.3. Okay, you will get 0.83823 exactly. But whenever you are going to multiply this with 100, you will get 82.3%. Okay, clear? Your phenotypic variance is equal to Vp is equal to Vg plus V. You should remember this formula. Okay, clear? Yeah, answer is B, that is 82.3%. When two species cannot be crossed directly due to incompatibility, a third species is used to make the target gene transfer possible. Again simple है, क्वेश्चन तो सिंपल है. बताओ, अभी बताओ, आंसर बताओ. Yes. See, breed species. Okay, everyone knows the answer is breed species. But if I ask you guys, okay, if I ask you guys example for breed species, okay, particularly in which crop we will try to use breed species more? Yeah, yes, tobacco is mainly used breed species. अरे भाई किसको यूज करते हैं टोबैको? नहीं नहीं, ऐसे एक क्रॉप को दूसरा क्रॉप के लिए एज ए ब्रीड स्पीसीज यूज नहीं करता है, ठीक है? वो तो डिस्टेंट हाइब्रिडेशन और वो पूरा अलग हो जाएगा, ठीक है? टोबैको 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 क्रॉप के अंदर हम रिलेटेड स्पीसीज को यूज करता है, वो तो ठीक है, मान लेंगे. बट व्हाट आई एम आस्किंग इज इन विच क्रॉप, ओके, इन विच फूड क्रॉप, okay, whenever I am asking the example you should always concentrate on major food crops, right? In which crop we are using more breed species to transfer wild genes from the cultivated species? अरे मैं तो बताया था, क्लास में भी बताया था, लाइव क्लास में भी. बताओ. अरे भाई wheat, ठीक है? In wheat we will going to use breed species since wheat genome contains A, B, B, D, D, okay, the multiple genomes. So you are not, it is very difficult to transfer some wild genes directly into Triticum meastypum. So what we will do in that particular case, first we will try to transfer our wild genes to tetraploid wheat, that is Triticum turgidum, A, B, B. After once we got the advanced line of AABV which is having the wild gene or any disease resistance or any quality traits from the wild relatives. So after that we will cross this Triticum turgidum with our wheat. Okay, we should remember this. Yeah, Ankur bata raha hai allopolyploids. Yes, exactly. You can see wheat is allopolyploid. Where there is an difficulty in crossing, okay, directly to the cultivated species, in that particular cases we will use breed species. Okay, the best example is we will use Triticum turgidum, okay, before crossing directly into the wheat crop. Merits of the pedigree method of selection in crop includes: it offers enough opportunity for natural selection to influence the population. It takes less time than the bulk breeding method. Breeders get maximum opportunity to use their skills. Transgressive segregants for yield and other quantitative traits may be recovered. बस एक ऑप्शन निकालना है इसमें. If you find that very early, then you can save the time. C, 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 A, C, D. ऑप्शन C लगा रहा है, A, C, D. It takes less time than the bulk breeding to develop an variety. ये सही है नहीं गलत है बताओ. क्यों आप B ऑप्शन निकाल रहा है? ठीक है, C नहीं है आंसर. D, D, D. B, C, D only. Exact answer is B, C, D. Okay, that means you can see I have told this in the previous question only. ऑलरेडी भूल रहे हैं आप, ठीक है? ऐसा नहीं होना चाहिए, ठीक है? तो ब्रीडिंग में ऑलरेडी बताया था 100+ मार्क्स गारंटी आपका जेब में है, ठीक है? In bulk breeding method, I already told that there will be enough opportunity for natural selection to influence, okay, on the population. What we will do after F2 and F3, we are trying to do bulk till F7, F8. Okay, we are not doing any selection in bulk breeding method, so that in naturally, okay, there will be some drought, there will be some heat stress. Okay, they will try to act upon the population. So only the resistant plants or tolerant plants will survive in that condition. That's only we will call it as a natural selection. ठीक है, कहाँ होता है वो? बल्क ब्रीडिंग में होता है, ठीक है? ये तो बल्क ब्रीडिंग में होता है. What we will do in pedigree method? After your F2, okay, we will try to select each individual F2 plants. Okay, you can use your skills there. Okay, by having some 2000 to 3000 F2 plants, you can select whatever you want by based on your judgment. Okay, that's what this statement is explaining. You can see breeders get maximum opportunity to use their skills and judgment for the selection of a plants, and transcription segments are the main feature of this pedigree selection. Okay, these are the three main merits of your pedigree breeding method. Clear? Bulk breeding method, two things you should remember. One is natural selection, another one is, बताओ, लगा दो कमेंट में, मैं बताया था already. What is the another merit of your bulk breeding method? One is there will be opportunity for natural selection. Another one is you can wait till the feasible environment. Okay, you can easily wait till the disease appearance, okay, or insect attack. That is the another advantage. The main similarity between the synthetics and composite. ले लो फिर से आ गया ये क्वेश्चन, बार बार आ रहा है, ठीक है, सब क्वेश्चन पेपर में आ रहा है, ठीक है, पक्का सिंथेटिक्स एंड कंपोजिट में एक क्वेश्चन तो आएगा. मैं ऑलरेडी बताया था प्रीवियस वीडियोस में हाउ वी विल डेवलप सिंथेटिक्स एंड कंपोजिट्स. Okay, just you have to choose answer now. The main similarity between synthetics and composites, both are relevant to allogamous species. It is right. This is the main output in cross-planted crop plants. Both can be reconstituted exactly and easily. ये सही है गलत है बताओ. कुछ लोग लगा रहे हैं ये, कुछ लोग लगा रहे हैं सी, ठीक है. पता नहीं वो सी कैसे आंसर हो जाएगा, ठीक है. योर आंसर इज ACD. ओके, विल सी. Both have broad genetic base, exactly right. Okay, as I told, both are heterozygous and heterogeneous. Both are mixture of several homozygotes and heterozygotes, exactly same. Okay, both can be reconstituted exactly and easily. This is false. Okay, which one can be reconstituted exactly? Synthetics or composites? Among these two, which can be reconstituted? Synthetic. Yes, Rimi. It is synthetic. Since we are having already tested for GCA, so we can use the same inbred lines again, so that you can reconstitute the genetic constitution. Okay? Yes. Good, Nutan. ये तो पॉलिनेशन सिंपल है आंसर. ये तो ornithopily, pollination by your birds, entomophily by insects. Okay, for A 3, for B 4, yeah, then C your hydrophily by water, C1. Okay, this is your answer. Clear? Actually this will be covered by your seed tech part, but it mixed up with our plant breeding slides. So I have seen this question also in so many papers. Okay, regarding again through the different pollination. Okay, they will definitely ask one question for you guys. Breeding a crop variety, okay, breeding a crop variety involves several activities. Arrange the steps of plant breeding program in correct chronological order. Again, it is very simple. Okay, they are asking these kinds of question very recently. Okay, in 2025 paper also they have asked the chronological order. Okay, since the pattern has been changed now, okay, they want to ask some of these statements kinds of question, so they will target these kinds of question. A, B, C, A, D. Okay, first what you have to do, first you have to collect, evaluate and selection of diverse genotypes for hybridization. Okay, first the germplasm collection is very important. Which are the target traits you are aims to develop. Okay, first you have to choose the target traits. Okay, for example, if it is disease resistance, you have to look for germplasms. Okay, then you can select some best donor parents, then recurrent parents. Okay, then you will cross among themselves. Okay, the second step is hybridization among selected parental genotypes. Okay, then you will going to select superior recombinants among those hybrid progenies. Then you will go for multi-location testing and release of a variety for commercial scale. Anyway, we already know that how the seed variety release chain will happen. Okay, starting from development, IVT, AVT1, AVT2, then identification, release and notification. Okay, your answer is BCAD. Next question. The development of a synthetic variety. ले लो फिर से आ गया synthetic variety. In a cross-planted crop involves several activities that are performed serially one after another. Again, chronological order. Okay, again it came in the last paper also, previous paper also. Okay, it is again, I think it is fourth time the question is appearing again. Okay, ले लो आप note it down. Synthetics, composites, hybrids. ठीक है, इसमें एक क्वेश्चन तो पक्का आएगा. So you should be very clear regarding the concepts. Okay, the different types of heterosis. Okay, mid-parent heterosis, economic heterosis, average heterosis, standard heterosis. Okay, you should know the heterobeltasis. You should know the each and everything about these things, formulas. Okay, then in heterosis there will be a different three phenomenon. Okay, like different three basis, genetic basis of heterosis, that is your dominance, O-dominance and epistatis. Okay, these are very important. Synthetics, composites and hybrids. Answer is BCAD. Collection of inbreds and testers. Yes, good. Then you will go for top cross or poly cross test for GCA and selection of inbred as a parental line. Yes. Okay, you should test for GCA in case of synthetics. After that, you will produce the synthetic variety. Then same as your breeding program, you will go for multi-location testing of that particular synthetic variety and distribution to the farmers. Your answer is BCAD. Clear. Very simple. The improved open pollinated varieties in maize have the following advantage. One thing you should remember here, okay? Open pollinated varieties are nothing but your synthetics and composites. In case of cross pollinated crop plants, they will try to make you confused, okay? If they are telling OPVs, okay, open pollinated varieties are nothing but your synthetics and composites. नूतन बता रहा है इनब्रिड विद गुड GCA. ठीक है, वो तो हो गया. अभी बताओ. What are the advantages of OPVs? Easier to develop than hybrids. Yes, it is advantage. Okay, we will leave for open pollination. Seed production is simpler and relatively inexpensive. Exactly correct. Okay, it is very simple seed production technology. There is no need for emasculation, crossing. Okay, there is nothing like that. Subsistence, farmers can save their own seeds for planting in the following season. Yes. Okay, since it is OPVs, okay, we can grow till some 3 to 4 years in synthetics. Okay, there is no need to replace seeds each and every year. Superior in yield than hybrids. Okay, this is incorrect statement. Okay, when it comes to yield, okay, your hybrids are superior. Okay, remember. Okay, note it down. A, B, D. A, B, D only. Your answer is B. Easy लग रहा है ना? क्यों easy लग रहा है बताओ? We are discussing these questions from past 4 to 5 classes. Okay, again and again I am explaining the same concepts so that okay, you are thinking that these are very easy questions. But if they ask somewhat out of the box, then you will get confused. Okay, but if you know the concepts very well, okay, then no need to worry in plant breeding. Yes. Okay, given below are the activities in the production of hybrid seeds of a crop. Find the correct group activities. Okay, in the production of hybrid seeds, okay, what are the activities they are asking? You should again you should remove one of the option. Okay, if you find that, then it's easy to choose. Then there is no need to invest your too much time. Okay, they are asking about hybrid seeds. Here they are telling about relative variance. Okay, निकाल दो B. किसमें B नहीं है? ये तो आपका आंसर है. Simple. Okay, you should be very fast in these kinds of questions. Effective generation and maintenance of male sterile plant. Yes, we need male sterile plants, and you should maintain male sterile plants for hybrid seed production. Production of F1 seeds through natural and manual pollination. Yes, you should go for GMS, CGMS, or any chemical hybridization agents. Okay, those are the different emasculation methods. And natural and manual pollination also. Sometimes we leave for insects. Okay, sometimes we'll do ant pollination. Okay, there will be a different natural and manual pollination, ensuring purity of the hybrid seeds through the space or time isolation. Okay, in single cross hybrid, in double cross hybrid, in three-way cross hybrid, we will going to maintain the isolations. Okay, बताओ अभी बताओ space isolation. Okay, our isolation for single cross hybrid, how many isolation we should maintain for single cross and double cross and three-way cross? Number of isolation बताओ अभी, ये भी important है, आपका CTEC में आएगा. There will be a possibility of this question. The number of isolation in single cross, double cross, and three-way cross hybrids. 7 for double cross, good. 4 for inbreed development, then 2 for single cross production, then 1 for double cross production. So there will be 7 isolation for double cross. Good, good. कौन बताता है? Ankur, yeah, good Ankur. Yeah, 7 for double cross. For your single cross, 3. Yes, Ankur. 2 for your inbreed A and inbreed B development, then 1 for hybrid production. Then three-way cross. In three-way cross, there will be 3 isolation for 3 inbreed development. Then 1 will be for your single cross hybrid. Then there will be another one for three-way cross hybrid. Okay, yes, good. 5 for your three-way cross hybrid. Note down. Okay, there may be a possibility of asking number of isolation for different hybrid seed production. 6 कौन लगा रहा है? नूतन, कहाँ से हो जाएगा 6? In three-way cross hybrid, okay, A into B into C. Okay, first to maintain A, B, C, there will be 3 isolation. Then to produce this single cross hybrid, 1 isolation. Okay, then again to produce this three-way cross hybrid, okay, 1 isolation. Total 5 isolation. ठीक है नूतन, समझ गए ना? C आउट ऑफ बॉक्स क्वेश्चन. ये तो एक और 2 क्वेश्चन ऐसे आएगा. They are asking about MABB, somewhat advanced. Okay, for that you should have some basic idea. Without knowing anything, you can't answer this question. Marker assisted backcross breeding. Okay, backcross breeding तो आपको पता है. ये marker assisted backcross breeding is a systematic approach to transferring a gene from a donor genotype to recipient variety of a crops. In normal backcross breeding also we will do the same, but here we are using the markers to see whether our gene is transferred or not. अब सी लगा रहे सब लोग पता है, सबको पता है. Here is the list of activities performed in standard MABB program. Production of F1 hybrids through hybridization between the donor and recipient genotype. Yes, same as your backcross breeding. In MABB also we will do the same. Then foreground and background selection and backcrossing. Yes, we will do this. We will do it. Hybridity, they are asking. Arrange the activities serially. Okay. First, what we will do, we'll select the donor and the recipient variety based on target gene. Okay. First, we have to use donor and recipient. Recipient variant should be mega variety. It is popular variety which is already growing in most of the areas. Okay. We just want to transfer any disease resistance or any target gene from the donor parent. Okay. First, you should select the donor and recipient, your D. Then after that, production of F1 hybrids through hybridization between donor and recipient parent, B, D, A. Then you should go for hybridity testing and backcrossing with the recipient parent. After you are crossing donor and recurrent parent, Okay, you will test for hybridity. That means your target trait from the donor variety is came or not. Okay, you have to test that. How you will test? Through the molecular markers. Okay, in MABB. That is your C. Then after that, foreground and background selection and backcrossing. Okay, after F1, you will go for backcrossing. Then you will go for foreground selection. Foreground selection is the thing, but by using markers, you will try to see whether your target gene is present or not. Your background selection is to see how much background genome came from the recipient parent. That is your background selection. That is your C, BA, DA, CB. DA, CB. मैं एक बात बताना था, ठीक है? मैं बीच में इसमें कुछ लेके आएंगे, मतलब क्वेश्चंस, इंपॉर्टेंट क्वेश्चन, ठीक है? उसको कौन आंसर करेगा उनको तो गिफ्ट मिलेगा मेरे से, ठीक है? आज तो नहीं है, कल से मैं पता नहीं किस क्लास में मैं लेके आएंगे, ठीक है? सब लोग लाइव क्लास अटेंड करने के लिए ट्राई करो, ठीक है? ओके, दिस इज MABB. कैन आई नो नाउ which is the first variety released through MABB? I have already told about this in your classes, live classes. बताओ. MABB is nothing but your marker state selection, which is the first variety in India. बताओ. इसको तो मैं गिफ्ट बताया था, वो गिफ्ट भी मिल गया स्टूडेंट्स को, ठीक है? One who answered for this question in my classes, okay, they already got gift. HHB 67, बस HHB 67 है क्या श्रुति? PHB 67, अंकुरदार बता रहे PHB 67. परमिलेट और क्रॉप तो पता है, परमिलेट में बस HHB 67 है क्या अंकुर? बताओ. बस HHB 67. अरे भाई मैं बताया था HHB 67 improved. HHB 67 is already popular variety. For that they have transferred downy mildew resistance genes. Okay, then it got improved as a HHB 67 improved. ठीक है, remember. And your improved Pusa Baspati 1, XA13, XA21 gene. Okay, these are the very important. Vivek sir had told this in the class. अरे अंकुर मैं भी बताया था, मेरा क्लास नहीं देखा है क्या? मैं पहली क्लास में बताया था शायद, ठीक है? ईयर भी बताया था 2005 and it is raised in 2006 in some of the other country, sorry not countries, other states, ठीक है? उसके बाद आ गया हमारा इम्प्रूव्ड पूसा बासमती 1, XA13 and XA21. उसके बाद तो there are so many marker tested varieties, okay, which are ruling at this particular time. Okay, one who are watching for the first time, okay, this is about our courses. One who are preparing for this present examination, 2026 examination, they are getting our course for 1,999. Okay, you can enroll and you can access our content. Okay, you will definitely get the so many questions from our courses. Okay, there is no doubt about it. For next year, our mock test bundle will be available at 799. Okay, and for 2027 course, okay, 4,999. And for 2028, our course price will be 8,999. Okay, as I am telling in each and every classes, okay, one who want to purchase our courses, you just take feedback from our already enrolled students so that you will get an idea, so that you can enroll to our courses. Okay, they are best mentors for you guys. Okay, your seniors or any one who is purchased our courses. Okay, you can just take a feedback so that you will get to know, okay, what the content we are giving and what are the different features of our courses. आ जाओ नेक्स्ट क्वेश्चन पे आ जाओ. रेसिप्रोकल रिकरेंट सिलेक्शन, ये भी मैं भूल गया था. ओके, हाउ दे आर आस्किंग synthetics and composites and hybrids, okay, 1 or 2 questions, they will definitely ask for recurrent selection also. Anyway, I have already explained recurrent selection in last class very clearly. Okay, I took some 10 minutes, so you can go and watch if anyone don't know about it. reciprocal recurrent selection simultaneously improves the two different populations in their ability to combine well with each other. Statement 2 बता रहा है इधर, your reciprocal recurrent selection is however inferior to the recurrent selection for GCA, recurrent selection for SCA when dominance is either complete or incomplete. कौन सा सही है इसमें बताओ? विशाखा, मधु हेगड़े, C. Okay, good. Your statement 2 is incorrect. Why it is incorrect? Equally effective. Okay. Equally effective or superiority? It is superior than recurrent selection for GCA, recurrent selection for SCA. बताओ. Equally effective, Sinchan. It is not inferior. Okay, in reciprocal recurrent selection, when you are trying to improve two population at a time, your one population used as a tester for another population, and population B is tester for another population, so that your reciprocal recurrent selection, when there is an either complete or incomplete dominance, okay, it is superior. Your recurrent selection for GCA will target additive gene action, okay, when there is a no dominance. Your recurrent selection for SCA will target over dominance, okay. So in reciprocal recurrent selection, you can achieve both, since your testers, that is your populations, okay, one for counterparts, they contains both homozygous and heterozygous material, so that you can test for both GCA and SCA at simultaneously. Okay, yes, superior, yes, good, Madhu. It is not inferior. This is wrong statement. So your third option is correct. Given below are the two statements. One is labeled as assertion and the other one is labeled as reason. Assertion is telling that the seeds obtained from an hybrid cannot be used for raising the next crop and need to produce fresh hybrid seeds for sowing in the next season. Your reason is selling selfing, segregation and inbreeding decreases heterosis in the seeds of hybrid crops. They have clearly given assertion and also your reason is correctly explaining the assertion. Okay, your first option is correct. When you will try to develop an hybrid, okay, for example, I am taking some three genes for example, okay, capital A, capital B, C. You will cross with small a, small b, small c, small c. You will get an F1, that is capital A, capital B, small b, capital C, small c. This is your F1. So this is hybrid. Okay. You will cross P1 into P2. Okay, the seeds, okay, the seeds will form in your female parent, that is in P1. Those seeds are your hybrid seeds. Okay, you will give those hybrid seeds to the farmers. Okay, whichever sets on the female parent, those are your hybrid seeds. You will give to the farmers. Farmers will grow those F1 seeds. Okay, when you grows F1 plants, okay, those are hybrids. Okay, the seeds will set on those F1 plants, right? Those are your F2 seeds. Okay, this is the condition of F1 plants. Since it is heterozygous and homogeneous, okay, whenever your farmer will grow, you will see the similarities in the field. At population level, it is homogeneous. Each and everything is similar, but it will show heterosis since it is in heterozygous condition. But whenever you will sow those seeds which are sets on the F1 plants, those are F2 seeds, when you will grow in the next generation, what will happen? This will be going to segregation. Okay, the possible phenotypes. Okay, what is the formula for possible phenotypes in F2? Batao. Formula for different phenotypes in F2. Yes. 3n. Array. Ankur. 3n or 2n? 3 power n. I am asking about phenotype. Rimi. अरे 4 power n गया. कौन? जंगम. अरे फिर से एक बार सोच लो. I am giving the second option for you guys. नूतन. अरे भाई क्या हो गया तुम? 3n for genotype. या I am asking about phenotype. नूतन, आप तो पहले ही बताया था क्या? या अरे भाई phenotype तो 2n है ना, 2 to the power of n. Okay, when in single gene, when you are considering single gene, tall and dwarf, how many different possible phenotypes in case of your F2? Tall and dwarf. Okay, if you confuse na, then you just calculate in your own mind for single gene, so you will get it. In case of tall and dwarf, there will be a one gene controlling it, so you will get capital A is small a small a. Genotypes will be 3, so there will be 3n. Phenotypes will be 2, tall and dwarf, 2 power n. Okay, no need to confusion. ठीक है, देखो आप सोच रहे हैं प्लांट ब्रीडिंग इजी है. इफ यू आर कंफ्यूजिंग, इफ यू डोंट नो द concept, then you will get confused here. So you can see here, when there are 3 genes are segregating, I am taking 3 genes. Okay, in F2 what will happen? The about phenotype 2 to the power of 3. 2 power of 3 is nothing but 8 in phenotypes. Okay, if farmers grows the F1 seeds, when your trait is controlling by 3 genes, you can see 8 different types of plants in their field. Okay, इसको हम बता रहे हैं segregation. Okay, it is nothing but segregation. ठीक है, समझ गया ना? So that for every year you should replace the seeds. Clear? बताओ कमेंट में बताओ. Is it clear regarding different phenotypes, different genotypes, why we need to replace the seeds? Yes. नूतन तो बस इमोजी लगा रहा है. नूतन का इमोजी पसंद है शायद. ओके कविता. ट्रांसजेनिक टमाटो वेराइटी फ्लेवर सेवर फिर से आ गया. Now I am telling, okay, again you will get one question from GM crops, maybe flavor saver, maybe your BT cotton, don't know from where they will pick the question. And again I am telling, this year maybe there is a chance to ask genome-edited rice. Okay, you should remember, they will definitely ask this particular question. Tomato flavor saver, greatly imposed shelf life. We are already discussed about it in yesterday's class. I have told each and everything. Okay, your antisense RNA technology. There is a gene for polygalactonose production. It will produce the 5-3, 3-5. This is your gene. It will produce mRNA. Okay, 5-3. When we will use antisense RNA, that means how we will use antisense RNA? We will reverse orientation of this particular gene. We will introduce into the crop. So you will get the reverse orientation. You can 3-5, 5-3. So you will get an mRNA again. That is 3-5. So it will correctly match us, since this mRNA is produced from this strand. Okay, this mRNA is produced from this strand. So when they will come together, complementary, and it will bind. So there will be a less production of your polygalactonose enzyme or complete absence of polygalactonose enzyme, so that your fruit life, the shelf life will generally increase. Kavita, DST, DRR rice. Yes, Kavita. That's what I have told. Pusa DST rice 1, DRR done. Yes, Kamala. Yes, good. Okay, the one who don't know about this particular genome-edited rice, I've already explained in our live classes. Okay, in marathon I will try to complete it. Cover it again. Okay, no worries. In standard PCR amplification with codominant marker, ये सिंपली कंफ्यूजन करने के लिए दिया है. Okay, whether it is for codominant marker, okay, for any normal SSR marker, okay, the standard PCR will need pair of primers, that is forward and reverse. In one cases, we will only use only one kind of marker as a both forward and reverse. In which marker? In which marker we will use only single primer as both forward and reverse. बताया था मैं, ये भी बताया था क्लास में. Which marker? इसको तो छोड़ दो आंसर. हाँ श्रुति, yes, for RAPD. ठीक है. In standard PCR we need both reverse and forward. In case of your SSR markers, you need both forward and reverse primer. In standard PCR, the statement 2 they are telling only the segment of DNA whose 3' ends are complementary to the 2 primers are used only gets amplified. ठीक है. What they are telling exactly in the statement 2, only the segment of DNA, okay, whose 3' end, that means you can see this is your DNA 5' 3' 5' sorry 3' 5', okay, will use primer. Where your primer will going to attach? Your primer will going to attach at this 3' end. Since you need 3' OH to continue the replication process. Here also it will attach here 5' to 3'. In statement 2, they are exactly telling that only the segment of DNA whose 3' ends are complementary to the 2 primers. 3' end, okay, the 3' end is complementary to the primer. Okay, then only you will get an amplified products. Okay, so what is the answer now? Answer बताओ अभी regarding 2 statements. मधु हेगड़े ये लगा दिया already. श्रुति ये. Yes, answer will be A. Okay, both are right options. You will get an amplified products. Okay, based on the number of cycle. Okay, 2 power n. N is number of cycle. Normally we will go for 30 to 35 cycle. So if you replace the 35 with the n, those are the number of fragments at the end of your PCR. ठीक है. सिंचो क्यों मैसेज डिलीट किया, रिट्रैक्ट कर दिया, क्या बता रहा था आप बताओ. Identify the correct statements regarding the mechanism and characteristics of agrobacterium infection in plants. ये भी कवर कर दिया मैं लाइव क्लास में. मैं कुछ नहीं छोड़ा है, मैं जेन्युइनली बता रहा है. मैं देख रहा हूँ ना क्वेश्चन पेपर. ठीक है, कुछ आउट बाउंड्री क्वेश्चन से कुछ ठीक है, कुछ मतलब बाहर से आ गया, मतलब ब्रीडिंग से बाहर, कुछ बायोटेक्नोलॉजी से, कुछ मार्कर से, वो तो सब कुछ कवर कर दिया ऑलरेडी. ठीक है, क्लासेस में तो ब्रीडिंग तो रिवीजन लिया था. ठीक है, वो भी कवर कर दिया मेरा हिसाब से ऑलमोस्ट. या आई एम स्लीपिंग सर. इफ यू स्लीप देन यू हैव टू स्लीप फॉर 1 more year. सिंचो ओके, आपका नाम तो बताओ, कल भी नहीं बताया था. हाँ, आंसर तो अच्छा से दे रहा है, बताओ आंसर, आपका नाम बताओ, गिफ्ट भेज देंगे. सी आई विल नॉट सिंग सर. अरे क्या बता रहा है तू, आंसर बताओ. सिंचन नहीं है, ठीक है, सिंचन. ओके, वेयर आर यू फ्रॉम? नूतन बता रहा डोरीमोन. डोरीमोन तो नहीं है. ठीक है, सिंचन अच्छा लड़का है. ठीक है, कहाँ से सिंचन? लड़का है, लड़का ही है ना, बिहार से, ठीक है, गुड। बताओ इसको आंसर बताओ। ACD रजत तो लगा दिया, ACD, ठीक है, वो आप ऑप्शन बता रहे, ठीक है ठीक है। ACD, अग्रोबैक्टीरियम काजेस क्राउन गॉल इन एप्पल, वो तो सही है, ठीक है। हाउ इट विल डू, I will explain in the next slide. Okay, how your crown gall will happen. C is agrobacterium in its gall by transferring tDNA. Exactly, it is right. Okay, your tDNA contains plant hormones. Okay, that is the main part of your TI plasmid. Agrobacterium need wounds to incite gall. Again, it is correct. Okay, agrobacterium causes gall by multiplying within the gall. No, it is not correct. Agrobac by within the gall, okay, there is no multiplication of your agrobacterium. Okay, it will indirectly it will transfer tDNA. In tDNA, it contains plant hormones. Okay, those are your axins and cytokinins. Those will cause for the repetitive multiplication of cells within that gall. Okay, your agrobacterium will not going to multiply in that particular case. Okay, so your answer is ACD. ACD. Because agrobacterium present in root, but disease occurs at plant part. That's what I am telling. It will just transfer. Okay, agrobacterium will just transfer the tDNA. So everything, okay, whatever happens after that, it will be in the plant cells. Your TA plasmid, okay, which is present in agrobacterium tumefaciens. That is your bacteria. It is independent, okay, self-replicating, okay, circular DNA. That's what your plasmid tells. The features of TI plasmid, it is around 200 to 250 kb length generally, but your tDNA region, okay, this is around 25 kb region. Okay, your tDNA content and your right and left border, those are 25 base pair repeats. It contains, your tDNA region contains auxin, cytokinin and opine. Okay, IAAH, IAAM, these are your auxins, and it contain IPT, that is your cytokinin, and it contains opines also. Those are nopalin. Okay, these are plant growth regulators. What we will do, oxygen and cytokinin, you know, anyway, basic. Okay, they will do the cell division. Okay, more and more, like in cancer cell, what happens, there will be accumulation of cells, okay, without proper cell division. Okay, like that in plants also, when this tDNA will transfer into plant cells, that area will try to multiply immediately, so that there will be a number of replications, number of cell division, your cells will increase and it will forms the tumor in case of apple. Okay, how it will happen, the procedure, okay, in simple way I will tell. Okay, for you guys there is no need to think about too much. Before that I will also tell, this is about tDNA region. There is origin of replication for the replication of TA plasmid, and it contains virulence region. This virulence region contains vir genes, vir A, vir B, vir C, D, E, F, G, H. ठीक है, ऐसे vir box है, उसमें so many genes are present. Okay, I will just explain simple way. Okay, first what will happen, this TA plasmid will be present in bacteria. Okay, when there is a wound in root cells in the apple tree, okay, what it will do, it will produce acetosyringine. So this chemical will stimulate this particular vir genes in the bacteria. First will be vir A. It will autophosphorylate. Okay, it will autophosphorylate and it will stimulate the vir G. This vir A will stimulate vir G. then this Virg, okay, it is like an promoter for all other genes, okay, activates complete Vir box. Vir box are nothing but your all other Vir genes. Okay, first you need wound, so acetosyringine it will produce, that will autophosphorylate Virya gene, then Virya gene will stimulates Virg gene, and this Virg will activates complete Vir box genes. Okay, these genes will, what they will do, simply they will cut this tDNA region and they will transfer into the apple cells in the plants, okay, where it got infected. Okay, after this transferred into the plant cell, plant genome, apple, for example, in this case apple genome, so they will try to do the unwanted cell division. ठीक है, क्लियर? इतना बस आपको इतना नॉलेज होना चाहिए. ठीक है, what Virya will do, what Vir G, what Vir, there will be so many genes again, Vir D, Vir E1, Vir E2, Vir C. Those are not important. Okay, at this point of time you should know only this basic idea. Acetosyringine, Virya, autophosphorylate, then it will activate Vir G, then it will activate all other Vir box. Okay, then the tDNA region will transfer to plant cells. It contains plant hormones, axin, cytokinin, and nopalin, opines. It will do the uncontrolled cell division. So you will get an galls. Simple. Second generation biofuels are made from, it is from economic botany, so I took this question. Okay, second generation biofuels. First you tell the first generation biofuels, then you should answer second generation. Yes. Come on. B. लगा दो, कमेंट पे लगा दो. आंसर बी है क्या? स्टार्च. अरे भाई हो गया तुम्हारा -5. आई एम आस्किंग फर्स्ट जनरेशन, देन यू टेल सेकंड जनरेशन. आप लोग कौन सा बता रहा है कमेंट में, वो बताओ पहले. आप लिख दो फर्स्ट जनरेशन ये, सेकंड जनरेशन ये केमिकल है, ऐसे लिख दो. Biofuels are very important. Okay, now we are targeting biofuels since you know the recent conflict for petroleum products. Okay, so biofuels are very important. Okay, first generation is starch, very good, starch and sugars. Second generation biomass, vegetable, potato. Okay, those are nothing but your lignocellulosic biomass. Okay, lignocellulosic biomass. ये तो बेसिक, इतना बेसिक तो होना चाहिए. ठीक है, लीव अबाउट सेकंड जनरेशन. फर्स्ट जनरेशन आल्सो यू शुड नो ना एटलीस्ट. अरे भाई, फर्स्ट जनरेशन biofuels made from food crops containing sugars, starch and vegetable oils. Okay, your maize, one of the important crop for biofuel production. Okay, it will produce ethanol from the starch. Biofuel are nothing but your ethanol. Okay, we can blend with the petrol. Okay, that ethanol will come from sugar and starch in case of first generation biofuels. But in second generation biofuels, it's made from the non-food feedstocks, primarily lignocellulosic biomass, such as your crop residues, okay, straw, husk and stalks from your wheat, rice, maize, forestry residues, energy crops. Okay, what are the advantages of second generation over your first generation? Do not compete with the food supply. If you use maize, sugarcane in the production of biofuel, then what about corn? What about sugar for the consumption? Okay, so in case of second generation biofuels, these are some crop residues and forest residues or any other energy crops. Okay, you can will not compete with the food supply and utilized agriculture and forest waste in case of second generation. ठीक है, third generation algae, fourth generation GMOs. ये तो, yes sir. Yes, good. थर्ड जनरेशन आगे वो तो मुझे पता नहीं, आई विल कंफर्म यू गाइस, ओके. सिंचन इस बता रहा है, पता नहीं, वो तो शायद जेन्युइन आंसर है क्या यार, ऐसे लगा रहा है कमेंट में. GMOs, we will not at present. I don't know, have idea about GMOs we are using for biofuel production. Okay, at this present current events. Okay, आगे तो एक बार देखना है. Yeah. What is process of obtaining regularity approval for use CRISPR-Cas9 technology in agriculture in India? CRISPR-Cas9 is nothing but genome editing. Okay. If it is SDN3, now it is changed. If it is SDN3, it will comes under GMOs. If it is coming under GMOs, that is nothing but CCC. Yes, Genetic Engineering Appraisal Committee, seeking approval from the Genetic Engineering Appraisal Committee. If it is SDN1 and SDN2, then there is no need to take permission from GEAC. It is normal conventional breeding. That's why we have released 2 rice varieties under genome edited, okay, as a conventional varieties. They will not comes under your GMOs. Approval or not appraisal, it is appraisal committee. Okay, you can see here guidelines for safety assessment of genome edited plants 2022. Recently they have changed, okay, the rules and regulation for the genome edited plants. That's why they have asked for this question in 2023. Okay, now recently they have released 2 varieties of rice. That's why maybe they can chance to ask those varieties in this particular this present exam. Okay, so you should be very careful. What it will exactly it will tell 2022 exemption. It is a landmark move from Indian biotechnology, from Ministry of Environment and Forest and Climate Change. Okay, issued a memorandum in March 2022 that accepted SD1 and SD2 plants, SDN1 and SDN2 plants from the stringent provision of the rules 1989. The original rules are 1989 rules. Okay, so from that rules they have exempted SDN1 and SDN2 from the stringent rules. Okay, क्या होता है SDN1 and SDN2? Repair mechanism. Okay, SDN1 is non-homologous end joining. Okay, there will be no addition or deletion. Sorry, there will be no addition, or you are not removing any part there. There will be a cut. Okay, by the phenomenon non-homologous end joining. Okay, there will be some addition of deletion of base pairs. Okay, there will be some mutations. But in case of SDN2, you are providing a donor parent to copy it. But in case of SDN3, that's why these two are exempted. Okay, we are not inserting any external genes. But in case of SDN3, okay, you are inserting an external gene. Okay, template DNA used, there is no template DNA used. Here we will use template DNA, yes, but it will not introduce into the genome. It will just, when there is a repair mechanism, when there is a cut, okay, it will just see this template and it will try to copy that sequence. But in case of SDN3, it is large template. Okay, here you can see small insertion or deletions or point mutation in SDN1. In SDN2, targeted substitution or precise small changes. But in case of SDN3, insertion of entire gene or a foreign gene. Okay, what is MOEFCC? विशाखा बताया था ना मैं, बताया था नहीं, Ministry of Environment, Forest and Climate Change. Foreign DNA in final product. There is no foreign DNA. Here also there is no foreign DNA. Here there is a foreign DNA. This is nothing but your GM crops, your SDN3. But in normal GM crops, okay, we don't know where exactly it is going and inserted. But in case of when you are using CRISPR, it is targeted modification. Okay, you know the target where exactly it is going and inserting. Regulatory status in Indian case, these two are exempted. As I told, you should remember This is regulated as GMO. Example, there are some, there is no examples in case of in wheat. It is not in Indian scenario. Knocking out allergen gene in wheat, aiding drought tolerance gene via SNP correction. Okay, introducing BT gene into rice. Okay, introducing BT gene into rice, that is through the SDN3. Okay, our Indian case, we have released two varieties. Okay, those are SDN1. Okay, our Indian genome-edited rice are through SDN1. This slide is going from the top, sir. Rajat, आप मतलब हमारा लाइव क्लास में नहीं था क्या? मतलब हमारा स्टूडेंट नहीं है क्या? The one who are from Agridic, they know this very well. I hope so. And for you guys, for you also, don't worry. Okay, this is some basic introduction. In marathon, I will try to cover simply that your CRISPR-Cas9. Okay, they will not going into the deep, but since it is in the news, okay, they may can ask in SDN1 and SDN2 and SDN3. Okay, which of the following molecular marker technique is used for identifying genetic variation based on the difference in DNA fragment size? I need exact answer. मार्कर आया तो आप सही से डायरेक्ट शूट करना है, ठीक है? इसलिए मैं सही से ऑलरेडी एक्सप्लेन कर दिया. मधु हेगड़े, रिमी प्रधान, ये. Okay, yes, RFLP. But what about SSR? SSR also there will be a different fragment size. Is it? What you will tell the basis of SSR then? If RFLP is difference in DNA fragment size, okay, the genetic variation based on the difference in DNA fragment size, then what about SSR? Rimi, Nutan, Ankur, या कविता बताओ, what about SSR then? Shinchan, number of tandem repeats. Ultimately, it is also some difference in fragment size, right? Some one band will come 400, another band will come 600 due to some repeats. How you can answer this question then, if they ask like this? बताओ. कितना हो गया टाइम? नहीं आ रहा है आंसर, ठीक है? If they ask like this, you should always go for RFLP. Okay, the genetic variation based on difference in DNA fragment size. But your SSR also due to different in fragment size, but they will specifically they will ask. Okay, the basis, genetic basis for SSR is due to number of tandem repeats. Okay, as Shinchan told. Okay, number of tandem repeats, that is your micro satellites. But ultimately your SSR also difference in fragment size. But the basic principle of your RFLP is difference in DNA fragment size after digestion with the restriction enzymes. Okay, अरे ले लो, ये तो बार बार आ रहा है, इस बार भी आएगा, ठीक है? नोट इट डाउन. The different international regulations and year of enforcement. First, I will start from CBD. 1992 is signed from all other parties, came into existence in 1993. Okay, your ITPG RFA, that is came into existence in 2004. Your next one is, I will take Cartagena protocol. This is also comes under CBD. 2000 signed, came into existence in 2004. Then your Cartagena protocol, sorry, your Nagoya protocol. signed in 2010, came into existence in 2014. Okay, what I left. Okay, these are the different years, okay, when they got signed and when they came into existence. कौन सा बता रहा है? C for A 4, yes for B 2, yes for Cartagena protocol C 1. Sorry, it is came into existence in 2003. Sorry for this mistake. And your Nagoya 2014. ठीक है, ये बहुत इंपॉर्टेंट है. And also you know the basic objectives, okay, the basic mandates. We can also tell that the basic objectives of this particular regulation, CBD, ITPG, RFA, Cartagena protocol, and Nagoya protocol. In yesterday's paper, I think, or day before yesterday's paper, they have asked about regulations and their objectives. Okay, you should know this so that you can get easy 4 marks in your paper. And your PPFR है तो ऑलरेडी मैं बताया था, कल में भी बताया था. ठीक है, छोड़ दो अभी. मैच द फॉलोइंग, ये भी सिंपल है. your Central Potato Research Institute is which is in Shimla. Your Indian Institute of Rice Research, which is in Hyderabad. Your NIPB, that is in Delhi, which is inside the IARI campus. Okay, your NIPB department, sorry, NIPB Institute. Indian Institute of Maize Research, which is Ludhiana. First it was present in New Delhi only. Now it shifted for Ludhiana. 3142. 3142. ठीक है, यस. कुछ एक्स्ट्रा इन्फॉर्मेशन दे सकते हैं क्या इसमें? Okay, anyway, you know NVP which is located in New Delhi again. Okay, these are some important institution. And you are, don't confuse with Indian Institute of Rice Research, that is IIRR, and Central Rice Research Institute, CRRI, and your International Rice Research Institute. Okay, your IARI is in Hyderabad, your CRRI in Cuttack, your IRRI which is in Manila, Philippines. There is one regional station of this particular IRRI for our region, okay, which is in Varanasi. Okay, regional station of IRRI in India, okay, which is presented in Varanasi. ठीक है, बैनलेट सिटी मनीला. वाह सिंचन, आपको तो पूरा जियोग्राफी भी है अच्छे से. हाँ, मेरा फेवरेट स्टूडेंट हो गया सिंचन. ठीक है, अंसुरी बता रहे हाउ मच टाइम फॉर कंप्लीशन सर. अरे ये तो लास्ट क्वेश्चन है शायद. सो रहा है क्या? जियोग्राफिकल इंडिकेशन इंक्लूड्स विच ऑफ द फॉलोइंग आइटम्स? जियोग्राफिकल इंडिकेशन इंक्लूड्स विच ऑफ द फॉलोइंग आइटम्स? बताओ, सब बताओ, जल्दी जल्दी बताओ, खत्म कर देंगे आज का क्वेश्चन पेपर. ए बी डी. Your old archaeological sites, structures, why we have to give for geographical indications for those things? Okay, your goods of handicraft, natural products, and agriculture produce. Okay, agriculture produce is the main core of the GI tag, okay, which is normally it will be applied from APEDA. Okay, since they will export our Indian agriculture produce to the different countries, okay, they will apply for generally for GI tags for different agriculture produce. Okay, and your GI Act, that is 1999. You should remember this also. Okay, this is about today's paper, that is 2023 shift 2. Okay, there are so many questions which are repeated, and they have asked some out of the boundary questions, and this paper they have targeted biotechnology and markers somewhat more. Okay, I hope that you are getting each and every questions, and for anyway one who are attending my live classes, they are feeling that yes sir, you have covered each and everything. Okay, लगा दो कौन कौन हमारा स्टूडेंट्स है, एक लाइक लगा दो कमेंट में, देखते हैं. कवर तो किया है ना मैं सारे क्लासेस में, रिगार्डिंग मार्कर्स, रिगार्डिंग बायोटेक्नोलॉजी, रिगार्डिंग सम क्वालिटी एंड डिसीज रेजिस्टेंस ब्रीडिंग। ठीक है, बस 3-4 बस उतना है क्या लोग कोर्स में, हमारा कोर्स में? श्रुति, यस, अंशुल, ठीक है ठीक है ठीक है, गुड गुड। ओके, नाउ वी हैव लेफ्ट विथ Shinchan. You are our master batch student or what? Just can you confirm once? Session is good, thank you sir. Okay, thank you so much. Okay, there are only 2 papers left, that is 2024 and 2025, recent 2 papers. Okay, we will do it tomorrow and day after tomorrow. I am not master batch. अरे क्यों मिस किया है आप? ठीक है ठीक है, अभी तो टाइम है, आप ले सकते हैं। नहीं हो तो आप अच्छे से करो। ठीक है, हमारा सारे फ्री कंटेंट है यूट्यूब में। ओके, हमारा वेबसाइट में भी फ्री कंटेंट मिलेगा, मॉक टेस्ट भी है। ठीक है, ले लो, अच्छे से प्रिपेयर करो। ठीक है, ओके देन वी विल मीट इन टुमारो सेशन विथ 2024 पेपर। ओके, टुमारो विच पेपर सर? 2024। ठीक है, या थैंक यू रेमी, थैंक यू सो मच। बट शिनचैन ब्रो, यू आर रियली गुड। केंद्रू बता रहे हैं शिनचैन इज रियली गुड। मॉक टेस्ट एंड एलाइट ग्रुप उसमें है क्या? अरे भाई, मेरा एक मैसेज डाल दो मुझे, ठीक है? ग्रुप में तो मैं भी है, एक मैसेज डाल दो उधर से। गुड नाइट सर, गुड नाइट। ओके, थैंक यू सिद्धार्थ, थैंक यू नूतन, थैंक यू वर्षा, गुड नाइट। वी विल मीट इन टुमारोज क्लास। ओके।